everyone. So today we'll be starting a new unit, that is diversity in living organisms. This unit has 3 chapters. First chapter is your biological classification. Then we'll be dealing with plant kingdom and animal kingdom, right? So let's start with the first chapter, that is biological classification. See, biological classification, as its name suggests, living organisms which are classified, that means biological, it means living organism has to be classified into different categories so that our learning becomes a little bit easier, right? It becomes easy for us to differentiate between living organisms, what is basically their physiology, how their body is working, what type of metabolism they are performing in, right? So for that reason, we are dealing with biological classification. Number one, that the very first question that arises is why we need to classify living organisms, right? Why we need to classify living organisms is the basic question that arises, but the reasons are very simple. See, we have seen so much of urbanization, right? We have seen so much of civilization, and with that civilization, we come across with new species, right? Old species are also there, new species are, we are come across new species. So we have to differentiate all those old and new species from each other. And for that, one important reason, classification is very important, right? Aristotle, he was the first who attempt to classify living organisms, right? Aristotle was the first the earliest attempt to classify living organisms was done by Aristotle, right? He hadn't given a more scientific reason. He actually classified living organisms on morphology, right? On simple morphology. That means on simple physical characters, on how the living organisms are looking, right? On their external features, he classified them into trees, shrubs, and herbs, right? On the basis of their height. Okay? Then he also divided animals into 2 groups which have red cell and the other who did not have red cell. It is written here. Animals with red blood cells like whales, fishes, birds, viviparous, oviparous, and animals who do not have red blood, that means insects, cephalopods, plants, and some animals as well. Right? So this is the actual reason why we need to classify living organisms. Let's come to the next topic, which is different type of classification. Now we are dealing with the other thing, that how many classifications are there. Right? We have started classification from 2 kingdoms. Right? In 1758, Aristotle, as we know that Aristotle didn't give any scientific reasons for classification, so that type of classification we didn't count it in scientific types of classification. We have the very first scientific classification was given by Carolus Linnaeus, and that was known as 2 kingdom classification. Carolus Linnaeus gave 2 kingdom classification in 1758, right? What did he say? He divided living organisms into 2 groups. One group is for animals and the other is for plant, right? We have plant kingdom and the other animal kingdom. Animal kingdom was known as Animalia and plant kingdom is known as Plantae, right? He classified these 2 groups on 4 bases. Number 1 is presence of cell wall, whether the cell wall is present in their cellular structure or it is absent, right? If it is absent, then they are animals, and if it is present, then they are plants. Type of nutrition, right? We all know that nutrition is of 2 types, autotrophic and heterotrophic. In autotrophic, autotrophic nutrition is shown by those living organisms who produce their own food. Autotrophs. Auto means self. Self nutrition is there. If they are driving self nutrition, then that is known as autotrophs. Plants, they are preparing their own food with the help of sunlight through photosynthesis, right? So they are known as autotrophs. And heterotrophs are the other organisms which depend on other organisms for their nutrition. Heterotrophs, that means they show different kind of modes for nutrition, be it saprophytic, parasitic, holozoic, right? be it any type of nutrition, all those they are coming up in one category, that is heterotroph, right? Heterotroph in very simpler language, these are the organisms which derive their nutrition from other living organisms, right? Third mode of classification is locomotion. Locomotion is that movement in which the organism is changing its position, right? Locomotion is that movement in which organism is changing its position. We have written here the difference between locomotion and movement, right? We have a very, very thin line difference between locomotion and movement, right? Movement is action that may or may not result in change of place. That means if we are standing still, if we are stationary and we are moving our hands or our head, then that is a movement, right? But if we are walking and which is resulting in change in our position, then that type of movement is known as locomotion, right? So that's why we have written locomotion here and not movement, right? So plants, they do not show locomotion because they cannot move, they cannot change their place on their own, but animals can. Animals can change their position by their own, right? The fourth mode of classification is response to stimuli. What is a stimuli? Stimuli is any change in surrounding. Any change in surrounding is known as stimuli, right? It may be external or internal change, anything which is changing around yourself, around you, and you are responding to that, that is known as your consciousness, right? So yes, there are a few plants who are conscious about their surrounding, but they don't have self-consciousness. Animals have self-consciousness, right? So this is the main criteria on which Carolus Linnaeus gives two kingdom classification, and this is the only first type of classification that we are dealing with, right? Then in 1969, five kingdom classification arrived. R.H. Whitaker was the one who gave this five kingdom classification. Along with Plantae and kingdom Animalia, he added three more kingdoms: kingdom Monera, kingdom Protista, and kingdom Fungi, right? So in total, he gave five kingdoms to classify living organisms, right? That is known as five kingdom classification, right? So there is a table in which this classification is shown, right? This five kingdom classification in which we have kingdom Monera, kingdom Protista, Fungi, and kingdom Animalia, right? So what is the actual basis of classification in this five kingdom classification? We have written the characters. There are in total five characters on which these five kingdoms are they are classified on the basis of these five characters. Number one, we have cell type. That what type of cell they have. We know that there are two types of cells, prokaryotic and eukaryotic cells, right? Prokaryotic cells are the primitive type of cells. Prokaryotic and eukaryotic. Prokaryotic is the primitive type of cell in which there is no membrane-bounded organelle present, right? But in eukaryotic type of cell, it is much more developed, advanced type of cell in which membrane-bounded cell organelles are present. Nuclear membrane is there, but in prokaryotes, no such structure is present, right? So let's see, only kingdom Protista. As you can see, only kingdom Protista is there which have prokaryotic type of cell. Rest four kingdoms, they all have eukaryotic type of cell, right? The other character is cell wall. The first question is the cell wall, whether the cell wall is present or not, right? And then if yes, then what composition of cell wall is there? Two questions are there for cell wall: whether the cell wall is present or not, and if yes, then what is the composition of cell wall, right? So as we can see that cell wall is present in starting four kingdoms, but it is absent in animals, right? We all know that. Into kingdom classification, we have also learned it that plants, they have cell wall, but animals, they don't have. That is the one important, classification basis that was used by Carolus Linnaeus. Again, by R.H. Whittaker, cell wall is used to classify kingdoms, right? So first we have Monera. Monera, let me tell you, kingdom Monera, it comprises bacteria, right? And bacterial cell wall, it is non-cellulosic. That means it is not made up of cellulose. It is made up of polysaccharides and amino acid. It has been written here. It's written here that non-cellulosic cell wall, it comprises polysaccharides and amino acids. Then we come to kingdom Protista. In kingdom Protista, we have different categories, right? When we'll be dealing with kingdom Protista, we will have that differentiation table in which different kinds of classes of kingdom Protista will be there, right? So in that case, you will be learning that different types of Protistants have different composition of cell wall, right? So yes, cell wall is present, but there is difference in classification, and some may also present in kingdom Protista which do not have cell wall, right? The other kingdom is kingdom Fungi. They have cell wall, and their cell wall is made up of chitin, right? Chitin is somehow a hard substance, right? So it is difficult to digest chitin, right? Then we have kingdom Plantae. Plantae, we all know that cellulosic cell wall is there in plants, which is very much difficult to digest. It is one of the most hardest organic matter present in earth crust, right? Then in kingdom Animalia, cell wall is absent, right? Next, we have nuclear membrane. This is somehow related to the type of cell, right? Type of cell, as I told you that prokaryotic cell, they do not have nuclear membrane, right? They do not have nuclear membrane, right? So that kingdom in which prokaryotic cell is there, it's but obvious that they will not have a membrane, nuclear membrane in their cell. It's somehow related, right? So only kingdom Monera is there in which prokaryotic type of cell is present. So nuclear membrane will be absent in them, and rest of the kingdom they will be having nuclear membrane, because the cell type in the next four kingdoms is eukaryotic, and eukaryotic cell, they have nuclear membrane. Right? Then next basis of classification is body organization. What is the actual body organization? That means the cells through which the body is made up of an organism, in what criteria, in what pattern they are arranged, right? So we all know that we have a cellular level, tissue level, organ system level, right? So let's start with kingdom Monera. See, as we go from a primitive type of cell to the well-developed type of cell, the organ level, it gets increasing. The complexity of body organization will get increased, right? See, prokaryotic cell, or you can say the kingdom Monera, which have prokaryotic cell, it has a primitive type of cell, so the cell organization will be simple. That is only cellular type, right? But as we go from kingdom Monera to kingdom Animalia, as the type of cell will get advanced or modified or developed, then this body organization will also get much complex, right? So kingdom Monera and kingdom Protista, they both have cellular type of organization, but from kingdom Fungi to kingdom Animalia, the complexity will get increased, and it will come up with multicellular, then tissue level or organ level, and then at last animals. We know that when we'll be learning this animal kingdom, no, then we'll understand that there are different type of phylums in which tissue level, organ system level is same, right? We have different animals, be it the simple animals or the complex animals. The criteria of body organization is different in different phylums of kingdom Animalia, right? So we have different type of body organization here. The last basis of classification is mode of nutrition. How they are getting their nutrition, right? We know that there are two main types of mode of nutrition, that is autotrophic and heterotrophic, right? We have learned that previously. So autotrophic, the kingdom Monera, they may be autotrophic, right? They may be autotrophic in which chemosynthetic autotrophs or photosynthetic autotrophic nutrition will be there. Yes, they are autotrophic, but on the basis of raw material they are using. Chemical, if they are using chemical for photosynthesis, if they are using sunlight for photosynthesis, then on the basis of this, they can be chemosynthetic autotroph or photosynthetic autotroph, right? Then they may be, some mangoes may be heterotrophic, right? In heterotrophic, there may be saprophytic, they may show saprophytic mode of nutrition or this parasitic mode of nutrition, anything, right? So the range varies, right? The mode of nutrition range varies in kingdom Monera because bacteria, no, as they are very simple in structure, but functionally they are very complex. This is the line of your NCERT only, that structurally bacteria are very simple, but functionally they are very complex, right? As we can see that functionally mode of nutrition is, it is very, it has different varieties. All the different varieties, they can be seen in one single kingdom, that is kingdom Monera, right? Then we come to the kingdom Protista, in which autotrophic and heterotrophic mode can also be seen, because I have told you that this kingdom Protista, it will be divided into different categories. 4 categories will be there. So we will be learning those in detail when we're dealing with Protista, right? Then we have fungi. Fungi compulsorily show heterotrophic mode of nutrition. They do not show autotrophic mode of nutrition. That means they cannot prepare their own food. They are completely dependent on autotrophs for their nutrition. So they can be saprophytic, which feed on dead or decaying matter, and they can be parasitic, which need a host, living host, for their nutrition, right? Then we have planted. You all know that plants are compulsory autotroph and they are photosynthetic autotrophs. That means they use sunlight for photosynthesis. Then we have heterotrophic nutrition in animals. Animals show compulsorily heterotrophic mode of nutrition. That means they cannot prepare their own food. They are completely dependent on different organisms for their nutrition. But which type of heterotrophic nutrition is there? It can be different, right? Because animal kingdom itself is a huge, it has different varieties. It's somehow a very vast kingdom in which different phylums are there. So there can be saprophytic mode of nutrition, holozoic or parasitic. Sorry. So we are done with 5 kingdom classification. So let's start with first phylum, first kingdom, that is kingdom Monera. Kingdom Monera comprises different kind of bacteria, right? So the sole members of kingdom Monera are bacteria. Sole members are bacteria, different type of bacteria, and they show extreme range in their habitats. From a very hot and humid climate, they'll be seen in very colder regions as well, right? So all the different kinds of habitats are written here: hot springs, deserts, snow, deep oceans, right? See how vast variety of habitats they are showing, right? Then the very first feature is they are present in different shapes, right? This round form is known as cocci, right? This somehow spoon shaped, right? They are known as bacilli, and spirala, which are somehow curved. They are known as spirala, and the last comma shape, they are known as vibrio, right? Comma shape are known as vibrio. So somehow this, right? So different kind of shapes are shown by bacteria. The other is respiration. Bacterial respiration is of 2 types. It may be aerobic or anaerobic. Aerobic, as its name suggests, it means in presence of oxygen. Oxygen is there, right? And anaerobic, in absence of oxygen. So bacteria are divided into 2 categories. One category is those bacteria who can respire in presence of oxygen, and the other are who can respire in absence of oxygen, right? Bacterial nutrition, if we talk about bacterial nutrition is of two types, autotrophic and heterotrophic. Autotrophic, somehow we have learned that autotrophic nutrition on the basis of raw material they are using can be of two types, photosynthetic and chemosynthetic, right? Photosynthetic are those autotrophic organisms or bacteria who use sunlight, right, for their electron transport or raw material. Sunlight is used. But in chemosynthetic, inorganic chemicals, inorganic chemicals as their raw material, right? They use inorganic chemicals as their raw material. Photosynthetic can be divided into further two categories, which are oxygenic and anoxygenic. Oxygenic, as its name suggests, it is in presence of oxygen, and anoxygenic, it is in absence of oxygen. That means photosynthetic bacteria prepare their own food in absence of oxygen and in presence of oxygen, right? Then we have other category, that is heterotroph. This is one of the most broad type of category in which saprophytic mode, parasitic mode, holozoic mode, different kind of modes can be seen, right? Let's come to the other feature of bacteria, that is bacterial reproduction. Bacterial reproduction can be of three types. See, bacteria, they don't involve in gamete formation and then their fusion. It's somehow just the transfer of genetic material from one bacteria to other. And for that genetic transformation, or you can say the transfer of genetic material from one bacteria to other, it can be transferred by three modes, right? First is binary fission. Binary fission, we know that when one single bacteria divides itself, its body into two bacteria, two individual, that is known as binary fission. Somehow it will be divided from center and then it will result in formation of two cells, right? This is binary fission. Then endospore formation. Endospore formation is somehow the process of spore formation internally. Internal spore formation is there. Internal spores are formed, right? Then last we have genetic recombination, in which just the genetic material is exchanged. Right? This genetic exchange can be of different types. It can be for transduction, it can be through conjugation or transformation, right? We'll learn all these different kinds of genetic transformation in higher categories, in higher chapters. Then we have pathogenic nature of bacteria. Bacteria can be pathogenic in nature, and then that means they causes disease, right? They cause different diseases like cholera, citrus canker, tetanus, typhoid. These are the four common diseases which can be caused by bacteria. Typhoid is caused by Salmonella typhi. Tetanus is caused by Clostridium tetani, right? So this is cholera is Vibrio. It is caused by Vibrio cholerae. So different kind of diseases are caused by different bacteria. That means let us sum up is that bacteria are pathogenic in nature, and that means they will cause different type of diseases, right? This is the completion of general features of kingdom Monera. Now let us come to the different categories of kingdom Monera, right? Kingdom Monera is divided into two categories, main two categories. One is eubacteria and the other is archaebacteria. See, eubacteria, they are known as true bacteria. It is very important. True bacteria. Why they are called as true bacteria, right? Why eubacteria are called as, only eubacteria are known as eubacteria, and the other categories are not known to be the true bacteria. Why? Because they have rigid cell wall. Rigid cell wall. This is the reason why eubacteria are known as true bacteria, because of the presence of rigid cell wall, right? And if motile, they may have a flagellum. A single flagellum is it can be there to provide motility to the bacteria, right? It comprises different members like cyanobacteria, which are known as blue-green algae. Okay, cyanobacteria are known as blue-green algae, and they are known as blue-green algae because they form water blooms, right? In polluted water bodies, they form water blooms. That means you'll see that the entire water body will become somehow greenish in nature, bluish greenish in nature, right? That is because of this presence of the cyanobacteria only, right? They are unicellular, filamentous, aquatic. Aquatic means they can live in fresh water as well as in marine, saline water also, right? And photosynthetic, they have chlorophyll A, which resembles the feature as of plants, right? This is the feature presence of chlorophyll A is their feature which resembles with the plant, right? It resembles with plant because plant also have chlorophyll A. And that is the reason why they are also autotrophic in nature, and plants are also autotrophic in nature. Then they are oxygen releasing, like nostoc, anemone. They have heterocyst for nitrogen fixation. Some bacteria, they fix atmospheric nitrogen, right, in form of nitrates or nitrites, right? And the examples of those are nostoc and anemone, right? We have one diagram of this nostoc also. This is the diagram of nostoc, right? We can see that the body of such bacteria who fix atmospheric nitrogen is divided into small colonies, right? Colonial form you can say. And within these colonies, a structure is also seen, which is known as heterocyst. This, right? This is the area for nitrogen fixation. Nitrogen fixation is done in this heterocyst only, right? This bluish color body structure is known as heterocyst, right? Then let's come to the other class of bacteria, which is archaebacteria. Archaebacteria, they are somehow different from eubacteria because their cell wall is not made up of, that it's not that made up of peptidoglycan, which is somehow the main composition, main component of bacterial cell wall, peptidoglycan, right? So their body is not made up of peptidoglycan. Instead, their cell wall is made up of polysaccharides, different sugars, and the polysaccharides and proteins also, which gives them somehow are resistant to harsh conditions, right? Their cell wall composition give them resistant to harsh condition. That's why they are so developed, or you can say they are so resistant to any alkaline, alkali, or any acidic, basic, or you can say any hot, humid, or very cold climate, right? They can resist extreme conditions because of their cell wall composition, right? So that's why they are somehow different from eubacteria, right? They can be halophiles, they can be thermoacidophiles or methanogens, right? Let us understand them one by one. Halophiles, somehow they are present in salty areas. Thermoacidophiles, they are present in areas which have high temperature, acidic conditions are there. Methanogens are those bacteria which are present in gut of ruminant animals and help in digestion of cellulose and responsible for production of methane or biogas, right? That's why they are known as methanogens, because they are producing methane gas or biogas in the gut of ruminant animals, right? Ruminant animals, they produce biogas and it can be seen through their dung, like dung cakes of cow dung is very much seen as it is used in form of fuel in rural areas, right? So why they are using cow dung cakes as a fuel in rural areas? Because it has methane gas, biogas, and that is because of these bacteria only, right? Yes. Then at last, kingdom Monera has another category which is known as mycoplasma. Mycoplasma, there are three features which are very important to remember about mycoplasma, and that is they don't have cell wall, right? They are the bacteria without cell wall. This is important. Let me star mark it for you because it is somehow very important point, right? MCQ can come from this. A type of bacteria which does not have cell wall, right? Then they are very small in size, pathogenic in nature, and they causes disease like mycoplasma pneumonia. And one more important point is this, that they can survive without oxygen, right? They can survive without oxygen. These are the 2 points which are very important, and they have been asked in previous years also. The type of bacteria which do not have cell wall and the type of bacteria who can live without oxygen, right? But statement based questions are also there, which are in the new trend. So you have to memorize all these statements about mycoplasma, because they can be asked like among these statements, which is the correct statement about mycoplasma or which is the incorrect statement, right? So statement based question is somehow a new trend. So we have to memorize each and every statement about mycoplasma, right? Let's come to the other kingdom, that is kingdom Protista, right? As we know that kingdom Protista, it comprises members who have eukaryotic type of cell, right? So they contain unicellular but eukaryotic organisms, right? They are unicellular, single cell will be there, single cell organism will be there, but that single cell is well developed. It is one primitive type of cell as compared to kingdom Monera. They have prokaryotic cell, which is a primitive cell, right? They have eukaryotic organisms, that means well developed cell will be there with nuclear membrane. Their boundaries are not defined. Primarily, they are aquatic, right? So let's start without wasting time. Let's start with the different categories of kingdom Protista. As I told you that kingdom Protista is somehow a huge kingdom which is divided into 4 to 5 categories, right? So we have in total 5 classes are there in kingdom Protista. Number 1 is chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Let's sum up this in one single table only. Let us first understand what type of members are present in these classes, right? Chrysophytes, they have members like dasmids and diatoms. Very important point to note, very important point. It has been asked several times. The dasmids, what are dasmids? What are they doing? What is their special feature? Right? So dasmids and diatoms are very important. Right? Members are dasmids, which are also known as golden algae, and diatoms, they are the chief producers of oceans, right? So in previous years, there has been a question that comes like that: what are the organisms which are known as chief producers of oceans? So diatoms. Diatoms are the organisms which are known as the chief producers of oceans, right? Then we come to the next class, that is dinoflagellates. Dinoflagellates, it contains members who are mostly photosynthetic, right? Who are mostly photosynthetic. That means they produce their own food. They are autotrophic in nature. Then euglenoids. Euglenoids show mixotrophic nutrition. Mixotrophic nutrition, that means they are autotrophic. They will behave as autotrophic in absence of oxygen, right? When there is an absence of oxygen, they will be work as heterotrophic, and in presence of oxygen, they will work as autotrophic. Why? How it is possible that in absence of oxygen they are working as heterotroph, but in presence of this oxygen they are working as autotrophic? Why? Because they have a pigment which is similar to those of plant, that is chlorophyll A, right? They have a pigment, chlorophyll A, they have chlorophyll A like pigment and acts as predators in absence of sunlight, right? That means in presence of sunlight, they will use their chlorophyll A like pigment for photosynthesis and will produce their own food. But in absence of oxygen, they will work as predator, right? Let's come to the other class, that is slime molds. Slime molds compulsorily they will have saprophytic members. That means the members who are feeding on dead and decaying matter, right? Protozoans, the members are heterotrophic. They show different forms of nutrition because we know that heterotrophic mode of nutrition comprises 2 to 3 classes of mode of nutrition. It may be saprophytic, parasitic, right, holozoic, different kind of heterotrophic nutritions are there. So they show different type of heterotrophic nutrition, right? Let's come to the other character, that is cell wall. Cell wall of chrysophytes, it is made up of silica, right? That's why it is known as diatomeous earth, right? When these organisms, the chrysophytes, when they leave their body, when they die, their cell wall, which is made up of silica, it deposits on the shore of oceanic bodies, right? And that shore region of oceanic body, it's known as diatomeous earth because it has silica, right? And that is used for different purposes, for filtration, right? Of syrups and juices and all. We'll be learning them in next slide also. Then let us understand cell wall of dinoflagellates. They have cellulosic plates on outer surface, right? They have cellulosic cell wall which is made up of cellulose. Euglenoids, they don't have a proper cell wall, but they have a special feature. They have a layer which is known as, they have a cellulosic, we can say a proteinous rich layer, protein rich layer, which is known as pellicin, right? Pellicin. This pellicin is very important, right? They don't have a cell wall, but they have a proteinous layer known as pellicin, right? Which gives them flexibility. So this can also be asked, that what feature is included in euglenoids with this pellicin or the proteinous layer. They are adding flexibility to them, right? Slime molds, they do not have a cell wall, but when they perform spore formation, when spores are made, when the process is going on, spore formation is going on, then the spores will have a true cell wall, right? Which is obviously happens in unfavorable condition. They perform spore formation, they do spore formation in unfavorable conditions, right? So in that case, they will have a spore wall, but not cell wall, right? And protozoan cell wall is absent. You can say that you can see that when we are going from chrysophytes to protozoans, no? If you notice that characters are characters are diverging from a plant character to a animal character. This is somehow it works. Right? The characters are diverging from a plant one to a animal one. Right? Then we have habitat. Chrysophytes, they found in fresh and marine water. They all are somehow the initial categories. They are the aquatic in nature. Euglenoids, they are present in fresh stagnant water, the still water, right? Slime balls, because they are saprophytic in nature, that means they will be living on dead and decaying matter. And because protozoans, somehow they have show heterotrophic mode of nutrition, so they can be parasite and this predator. If they are parasite, that means they will live on the host body. Next, we have examples and the special features, right? Special feature for chrysophytes, as I have told you that their cell wall is made up of silica, and that means they will form a diatomeous earth, right, which is gritty in nature and used for polishing and filtration of oils and syrups, right? Dinoflagellates, they have different color pigments, so they can be of different colors. They can be yellow in color, brown, green, or red in color on the basis of presence of pigment, right? The most dangerous one is red algae, and the example is Gonolex. Gonolex is a red algae which is very dangerous in nature because it releases a toxic which is very dangerous for the aquatic lifestyle, be it plant or other smaller animals. Euglenoids, they are the connecting links between plants and animals, and their special feature is that they have two flagellars. One is the smaller one and the other is large one, right? Somehow like this. One small one and the other will be the large one, somehow like this, right? Then the special feature of slime mold is that in favorable conditions they will grow without dividing to several feeds, and that structure is known as plasmodium. But in unfavorable conditions, they will form spores which have true spore wall, right? They will have true spore wall, right? And these are dispersed by air current for fertilization or pollination. Then protozoans. Protozoans are somehow divided into 4 categories. Number 1 is amoeboid. They have pseudopodia. The special feature is that they have finger-like projections which are known as pseudopodia. That means false fingers, right? They use this pseudopodia for food engulfing, food arrangement, everything, right? Then we have flagellated protozoans. Flagellated protozoans, they are specifically free-living or parasite, and the example is trypanosoma, that which causes sleeping sickness. They causes a disease which is sleeping sickness. Next we have ciliated. Ciliated means that they will have cilias on their entire body, and they will function as they will give a movement, coordinated movement, in order to give a overall movement to the bacteria. And the example is paramecium. Last category is sporozoans. Sporozoans have all the sporozoans, they will have one infectious spore stage in which they will be causing any disease, and that is plasmodium, which causes malarial disease, right? Malarial parasite, plasmodium. So with this, we have completed kingdom Protista and kingdom Monera. So let's start the other kingdom, that is kingdom fungi. We have written some specific features of kingdom fungi. They are heterotrophic organisms. They are also when we are talking that when they live in association, we know that they live in association with algae, right? So why they live in association with algae? Because algal partner will provide food to them and they will provide shelter, right? So that's how we conclude that fungi are heterotrophic in mode of nutrition. They don't prepare their own food. Their hyphae is somehow thread-like structures like this. Thread-like structures are there, right? And they're entangled together, right? These structures are entangled together, right? When a network of hyphae, as we can say that this is an entangled structure of multiple hyphae, so this is known as mycelium, right? A single thread-like structure is known as hyphae. Single thread-like structure is known as hyphae, and the complete network of hyphae, multiple hyphae, is known as mycelium. The mycelium, they are multinucleated. That means they have multiple nuclei, right? Which is also known as cenocytic hyphae. Cenocytic hyphae, right? Their cell wall is made up of chitin. We have read this in table as well, classification table as well. Their cell wall is made up of chitin and polysaccharides. They are cosmopolitan. That means they can grow anywhere, especially in warm, hot and humid areas, right? Damp places. They grow in especially damp places, right? Let's come to Fungal mode of nutrition. Fungi shows, because they are heterotrophic in nature, so they specifically show saprophytic mode of nutrition and parasitic mode of nutrition, right? They live in symbiotic association with an algal partner or with the roots of higher plants. When they live in association with algae, that is known as lichen, and when they live in association with roots of higher plants, then that is known as mycorrhizal. With roots of higher plants. Right? So they can be saprophytic in nature and they can be parasitic in nature. And the third mode of nutrition is that they live in a symbiotic association with the other partner, so as the other partner will provide food to them and they provide shelter to that association, right? Example, Buccinia, which is also known as Beetrus, penicillin yeast, which is a unicellular fungus. Then we have fungal reproduction. Fungi reproduce through different modes, right? They can reproduce through vegetative formation. Vegetative propagation is there, right? In form of fragmentation. Fragmentation is that type of reproduction in which a single fragment can develop a complete individual, right? Fission is there, binary fission or multiple fission. Budding is there, that means an external bud will grow on the body of fungi, and then that bud will detach from the body and will grow into a new individual. Then we have asexual and sexual mode of reproduction. In asexual, both sexual and asexual mode of nutrition, it has been done by spore formation, right? Asexual spores are there and sexual spores are there. Asexual spores such as conidia, sporangiospores, right? Zoospores, and sexual spores are oospores, ascospores, basidiospores. You have to memorize the name of these asexual and sexual spores. The question they can simply give you through this on this different types of spores. You have to recognize that what particular option is of asexual spores and the other is of sexual spores, right? So you have to memorize the name of sexual and asexual spores. Then fruiting bodies are involved, obviously, right? Before spore formation, they develop a structure which is known as fruiting body, and on those fruiting bodies, spores are formed, right? So spore formation is only possible when fruiting bodies are there, right? So fruiting bodies is an initial step of spore formation, and then spores will be formed whether it is a sexual spore or a sexual spore. Okay, got it? Then let's come to sexual reproduction. Sexual reproduction in fungi, it's somehow different, right? It contains 3 steps. First step is plasmogamy, karyogamy, and meiosis. Let us understand what these 3 steps say. Plasmogamy is the fusion of protoplast. Protoplast is somehow the material inside the cell, right? Matrix. It is the cell matrix, right? Protoplast is cell matrix. Right? Right? So plasmogamy, when the protoplast fuses, that is known as plasmogamy. Then karyogamy, it is the fusion of nuclei, and then meiosis, the formation of haploid spores. So these are the 3 steps which are involved in sexual reproduction of fungi. Plasmogamy, karyogamy, meiosis. Plasmogamy is fusion of this protoplast, karyogamy is fusion of nuclei. That means first of all the matrix will get fused. If 2 cells are there, they are performing sexual reproduction, they are in the process of fusion. So their protoplast will fuse first, then the nucleus will get fused, and then meiosis will be there. That means the chromosomal number will get half, right? To form haploid spores, right? Related to this sexual type of reproduction, we have one different stage that is known as dikaryon phase. Dikaryon phase, it's somehow that stage. See what happens in some fungi, plasmogamy and karyogamy, they are not followed immediately by each other, right? Plasmogamy is followed by karyogamy after a minute gap of time, right? And that gap in which that multinucleate situation is there, right? We say that in plasmogamy, protoplast has been fused, right? So that means there are two nuclei present inside that one protoplast, right? This stage will continue to go for a very short period of time before karyogamy, right? So in some fungi, plasmogamy is not immediately followed by karyogamy, right? There is a difference, there's a time period gap is there between plasmogamy and karyogamy. That multinucleated situation in which two nuclei are present in one cell, that is known as dikaryon phase, right? And this is found, not all fungi shows such type of intermediate stage. Only two categories of fungi, Ascomycetes and Basidiomycetes. These are the two categories of fungi which shows this intermediate stage of sexual reproduction. Rest, there is an immediate, follow-up of karyogamy of plasmogamy by karyogamy, right? Let's come to the classes of fungi. Two we have learned here only, Ascomycetes and Basidiomycetes. Other two are Phycomycetes and Deuteromycetes. Let's come to the first type of fungi, first category of fungi is Phycomycetes. See, the only important thing that you have to remember about different classes of fungi, it's the type of spores they are forming. What type of sexual and asexual spores they are using, right? Then Phycomycetes, they grow on dead wood, dead or decaying matters. They can be parasite, they can live on host body, and they can also live on dead or decaying matter, right? The asexual spores are zoospores and eplanospores, and sexual spores are zygospores. Example, Rhizopus, albigo, and mucor, right? Examples are very important, right? Question will be given you that which fungi uses eplanospores for this asexual reproduction. Then you have to choose between these three, right? Then we have Ascomycetes. Ascomycetes are also known as sac fungi, and they have asexual spores as conidia and sexual spores as ascospores, right? And the examples are Aspergillus, Neurospora, Saccharomyces. Saccharomyces is the other name for yeast, right? We have heard yeast so many times. The biological name for yeast is Saccharomyces. It's a unicellular fungi. Claviceps, morels, and truffles. Morels and truffles are the edible fungi. These are the edible fungi. We can learn it as we eat the truffle pastry, chocolate truffle is there. So truffles, you can remember them somehow like this. So truffles and morels are edible fungi. Then Basidiomycetes, they are septate and branched mycelium. That means their mycelium will not be, unbranched. They are branched, they are divided, right? Sexual spores are Basidiospores, and the fruiting body is Basidium. This Basidium is fruiting body. We have discussed that what are fruiting bodies, right? These are fruiting bodies. Asexual spores are absent. So this is one of the important points for Basidiomycetes, that they do not reproduce asexually because asexual spores are absent, right? Examples are Agaricus, Eustelago, and Puccinia. Then last we have Deuteromycetes, which is also known as imperfect fungi, right? Imperfect fungi is because their sexual stage is unknown. Sexual spores are not there, right? Sexual reproduction is not there. So that's why they're known as fungi imperfecta, right? They are, if the sexual spores has been somehow, in future, if the sexual spores are identified, discovered in Duromycetes, then they will be classified as Ascomycetes or Basidiomycetes because they have so common features, right? Their examples are Alternaria, Trichoderma and Trichum. Okay? Coleto, So this completes your kingdom fungi. Now the last part of your chapter is viruses and viroids. What are viruses and viroids? See, there's a debate going on from very past years that whether the viruses are living or non-living. Let me tell you that viruses, they are non-living because two points are there. One important point is that they do not have plasma membrane as like the other living organisms, and secondly, they are living only when they enter the host body, right? Because once they enter the host body, they start multiplying themselves, right? They start replicating themselves. But before that, when they are not inside a host body, they are living on their own, then in that case, they will not be multiplying, replication is not there. So there are the 2 points through which you can understand that viruses are non-living. First is they do not have a plasma membrane, and other, they divide only when they enter into a host body, right? So viruses, it's non-cellular, inert, outside host, right? Firstly, Pasteur was the one who coined the term virus, and it was discovered by Ivanovsky, right? So you have to remember the name of the scientist who discovered virus, Ivanovsky, and who coined this word virus, Pasteur, right? They are the 2 different scientists who discovered and who named it as virus. Then it is Vigernik used one statement, that is contagium vivum fluid. That means it is a fluid through which it uses viruses. They uses a fluid which is contagious in nature. That means which is disease causing, right? So this contagious vivum fluid, this is the statement given by Vigernik, which says that viruses have a fluid which is contagious in nature. That means they are disease causing. They are pathogenic in nature. And Stanley crystallized them. To know their structure more easily and deeply, it has to be crystallized. And who crystallized it? Stanley crystallized it. Okay? Now let's come to the structure of virus. You just have to understand that what are the different genetic compositions are there. Some viruses, they have DNA and some may have RNA, right? And also DNA and RNA can be single-stranded and double-stranded, right? Plant viruses, they have single-stranded RNA, right? Bacteriophage. Bacteriophage are those bacteria there who infect bacteria only, right? Viruses who infect bacteria are known as bacteriophages. They have double-stranded DNA, and viruses who infect animals, they can be single-stranded RNA, double-stranded RNA, or double-stranded DNA, but they cannot be single-stranded DNA, right? So these are the 3 important points about the virus structure that has to be memorized, right? Then they are made up of a protein layer, and that protein layer, they are made up of small capsids, right? See, capsomere is somehow like this. Small structures will be there, right? These are the capsomeres. Capsomeres, right? These are the capsomeres. And these capsomeres will form complete capsid, which is a proteinous layer. It is one of the important feature of virus structure that they will have a protein layer which is known as capsid. Right? Let's come to the diseases which are caused by virus. Virus cause different type of diseases in humans, plants and bacteria. If we talk about humans, then mumps are there, right? Is a very common disease, right? That parotid glands, salivary glands, they get swelled up because of the viral infection. Smallpox herpes, influenza, AIDS. They are the main viral diseases which are very dangerous. In plants, it causes mosaic formation, leaf rolling. Leaf will get rolled up, right? So that they cannot perform photosynthesis properly. Leaf rolling is there, yellowing of leaf is there. That means chlorophyll A, the pigment, will get reduced. Dwarfing, small size of leaf or plant, stunted growth. That is also a kind of dwarfism only, right? That means the growth of plant will get hindered. Next, we have viroids. Viroids are the infectious agents which have free RNA, right? They are the infectious agents which have free RNA. Somehow they are smaller in size than virus, right? They are smaller in size than virus. Smaller in size than virus, right? And they causes potato spindle tuber disease. They are discovered by T.O. Diamond, right? Then at last we have lichens. Lichens, as I have told you earlier, that this is a symbiotic association between algae and fungi, in which the algal partner will provide food because they are autotrophic in nature. So they will, through photosynthesis, they will produce food. But the fungal partner will provide shelter to them, right? So both of the partners, they are providing each other different necessities. That's why they live together in a symbiotic association, and that association is known as like. It has been written here. It is written here that algal partner will provide food, right? And fungi provide shelter and absorb nutrients, right? One of the important point is that they are pollution indicators. That means lichens are not found in those areas which are more polluted, right? Where there is more pollution, lichens will not be present in those areas. So they are considered as pollution indicators, right? So this completes your chapter biological classification, right? The question that generally arises is among the classification system, right? Be it Protista, kingdom fungi, these are the two things, and also kingdom Monera. These are the three things which are very important to memorize, right? From kingdom Monera, kingdom Protista, and kingdom fungi. There are different kind of questions that can arise, and the only thing that you have to memorize is the classification characters, that on what characters the classification has been done, right?